Okay. Okay. Let's go through multiple choice for test number 10. First one. A genetic counselor worried about developing Huntington's disease, an inherited disorder caused by a dominant allele of a single gene. The young man explains that his cousin was recently diagnosed with Huntington's disease, and the news has caused him to consider his own risk of developing the disorder. Which of the following questions will best help the genetic counselor to evaluate the risk of the young man developing Huntington's disease and transmitting it to his children? Okay, so I mean, basically, um, his cousin got it and he wants to know if he has a chance, right? So any question related to family history, right, would work, right? So B, were your parents or grandparents ever diagnosed with Huntington's disease, right? Remember, Huntington's disease is one of those that are homozygous lethal, right? It is a dominant disorder. Um, you cannot have two copies, right? Um, yeah. Okay. Over several years, bacteria were isolated from members of a human population and tested for antibiotic resistance. The percent of bacterial isolates that were found to be antibiotic resistant is presented in the graph above for each year of the study. Which of the following conclusions is best supported by the information presented in the graph? Okay, so um, you can see over like what course of 8 years, right? Looks pretty much close to what it goes from like maybe 2%, right, to about 20, right? So you have kind of rapid growth, right? You don't see much change until probably like year 3, and then from year 3 you kind of see like an accelerated exponential type curve, right, in terms of the percentage of resistant bacteria, right? Okay, so which of the following conclusions is best supported by the information presented in the graph? Greatest increase in antibiotic resistance occurred from the first year to the third year. Not true, right? You can see that there's pretty much no change. Gene flow in the population occurred at the same rate for the entire duration of the 8-year study. This is not what this graph shows, right? Yes, there could have been gene flow, right? But that wouldn't be this. That couldn't be, I guess, explained by this graph. Because evolution in bacterial populations occurs over millions of years, the results are due to random variation. This is also not true. Remember, bacteria is one kind of, because they uh, reproduce so rapidly, right? You can see, right, natural selection occur in bacterial populations where you normally can't see it in other living creatures, animals, organisms. Like for example, like um, I don't know, if a species of birds, right, whatever, were undergoing natural selection, really hard to see, given that it takes such a long time, right? So your one exception to that would be for bacteria. Okay, natural selection over multiple generations is favoring antibiotic resistance in the population of bacteria. Yes, right. Oops. Right. So. So D, right? You can see that there is obviously selective pressure for antibiotic resistance, right? That's why you see a change in the percentage. Okay, lobe-finned fishes were present in the oceans of the world approximately 400 million years ago. The first tetrapods, vertebrates that had limbs and could move on land, date to about 365 million years ago. One hypothesis states that early tetrapods evolved from lobe-finned fishes. Which of the following is the best plan for testing the hypothesis? Okay, um, so basically, right, this theory, right, how would we test the theory that tetrapods evolved from lobe-finned fishes, right? First one, determining the number of fins found on lobe-finned fishes from different geographical locations. Again, you're just comparing lobe-finned fishes, right? So how would that would not, right, help in drawing a conclusion between any kind of that the what is it called? Lobe-finned fish is a ancestor, right, of oops, tetrapods. Okay. Measuring the proportions of radioisotopes found in fossils of early tetrapods. Again, that would help you in dating. Yeah. Okay. That would help in dating the fossils, right? So how old they are wouldn't tell you much more, right? Comparing the arrangements of bones in the fins of lobe-finned fishes and limbs of the earliest tetrapods. Um, Yeah, right. If you look at bone structure, right, you can search for kind of homology, right? Again, that would give you a hint as to whether they have shared ancestry, right? Sequencing DNA isolated from fossils of lobe-finned fishes to determine the size of the genome, right? You can use DNA sequencing, right, in order to, if you wanted to compare um, whether there were significant similarities between tetrapods and lobe-finned fishes, but doing this part, what they describe in D, is not useful. So C. Which of the following describes a metabolic consequence of a shortage of oxygen in muscle cells, right? So what happens when we run out of oxygen, right? Your muscle cells can no longer do cellular respiration. If your muscle cells can no longer do cellular respiration, what is going to happen? They're going to have to resort to fermentation, right? So your oxygen is going to do fermentation, and what are we going to produce? We're going to produce lactic acid. Right? Um, yeah. Remember, lactic acid is like um, responsible for like when you're running, right? And then you get like that sharp pain in your muscles, right? Like your legs or whatever. That is due to a buildup of lactic acid. Okay, prairie dogs are small animals that live in large colonies in burrows in the ground. Prairie dogs that are near their own relatives when a predator approaches are much more likely to issue a warning bark than those that are near unrelated prairie dogs. The prairie dogs that hear a warning bark are more likely to hide in their burrows than to remain above ground. However, the prairie dog that gives the warning bark is putting itself at increased risk of being identified and killed by the predator. Which of the following presents the most likely evolutionary explanation for the behaviors described? Okay, so again, right, um, what kind of, or I mean, yeah, um, let's kind of go through. Warning bark changes the behavior of the related prairie dogs nearby, allowing the prairie dog's family to have increased survival and reproductive success. Right, this is very likely what is happening. Right, that warning bark. Right, if you think about it, um, it puts itself at risk, right? However, why would it still issue that warning bark? Um, it's because it's trying to essentially, right? at least help the other, right, related prairie dogs have a greater chance of survival, right? Um, yeah. Again, barking prairie dog chooses to warn other prairie dogs, leading to more prairie dogs living above ground. Yeah, not an evolutionary explanation. Barking prairie dog is alerting unrelated prairie dogs to the predator. That is contradictory to what they just said in the little blurb. Failure of the individual to bark when surrounded by unrelated prairie dogs ensures survival of the individual. Doesn't necessarily ensure survival. Yeah, it can increase it greater, right? But this explains why it um, doesn't do something, right? So A is your best answer. Okay, number 6. One model of a sustainable fisheries practice is for individual fish to be removed from a natural population at a rate equal to the highest possible growth rate of an ideal population. The graph above represents a population of bluefin tuna living along the Atlantic coast. At which labeled point in the graph is a population growth rate the highest? Okay. Um, so basically, right, um, it's just tracking population of bluefin tuna, right? Where do we have the largest population growth rate, right? Population growth rate is just change in your population, right? So it's going to be part 3, right? And this kind of looks like our logistic growth model, right? Our sigmoidal curve, right? Where this right here, somewhere over here, would represent your carrying capacity, right? Here you have kind of this period, right, of rapid growth, right, where if you kind of just look at it from here, it looks exponential, right? But remember, anytime your population approaches your carrying capacity or gets closer to your carrying capacity, you have deceleration in your growth rate, right? Or meaning your growth rate is just going to slow down. Because again, what is carrying capacity? It's the maximum number of individuals that your environment can support, right? So answer 6 is C. All right, effect of water-soluble pollutants on membrane permeability. A student formulated a hypothesis that water-soluble pollutants damage living organisms by increasing the permeability of cellular membranes. To test the hypothesis, the student investigated the effect of isopropanol and acetone on beetroot cells. The vacuoles of beetroot cells contain large amounts of beta-cyanine, a water-soluble pigment that is released into the extracellular environment as a result of increased membrane permeability. The student prepared identical samples of beetroot tissue and incubated each sample for 15 minutes in the specific solution for that group. At the end of the incubation period, the student measured the absorbance of 460 nanometer light for each sample. A greater concentration of beta-cyanine in the solution surrounding the beetroot cells results in a greater absorbance of 460 nanometer light. The results of the experiment are shown in the table above. Okay, so what exactly is going here, right? So basically, their idea is that these pollutants, right, um, are harmful by increasing solubility, right? Meaning that things that should or should not leave your cell are are doing it, right? Causing cell damage, right? Um, so like the vacuoles of beetroot cells, right, is what they have here. So beta cyanine, right? Um, and then you looked at absorbance, right? 460 nanometers, right? Absorbance, right? How much light is absorbed? How much is deflected, right? Okay, which of the following is a dependent variable of the experiment, right? So you have your independent variable, which is what you manipulate, right? Dependent variable are your results, right? So if you look here, right, what are we manipulating? Treatment solution, right? So in this case, our dependent variable is the percentages, right, of isopropanol or acetone, right? And then what are we measuring, right? We're measuring mean absorbance of 460 nanometer light, right? So treatment solution is our independent variable, mean absorbance of light is our dependent variable. So the only one that, oops, D, absorbance of light. Okay, which of the following graphs is the most appropriate representation of the experimental data, right? So we have kind of like this, right? How would we kind of show that, right? We have bar graph, line graph, pie chart, and this, right? So again, remember, anytime you're graphing your data, right? X-axis is always independent variable, Y will always be dependent, right? Let me see. Obviously, right, these two, C is definitely not how you want to graph it, right? Pie chart is definitely not the way to go. If you look at D, right, D and A are essentially the same thing, right? Problem with D, right, is they didn't add standard deviations, right, or your error bars, right? Anytime you are given data, especially like in this case where they literally give you, right, 2 times standard area of your mean, right, and you did not add error bars, yeah, you should always add your error bars. So in this case, right, line graph is the best way to go. Bar graph doesn't necessarily work here as well, right? Because we're trying to see a trend, right? When you change your percentages, right, what happens to your absorbance, right? So the best way would be with a line graph. So 8. Actually, A A is probably a better answer because the treatment 1, 2, 3, 4, and 5 are not related, right? You see the data. Yeah. Separate groups, right? Line graph, if treatment groups 1, 2, 5, right, it was like connected somehow, right? Or like it was some kind of like progression, right? Like 0 to, or I mean, not necessarily, but like 0 to 10 seconds, right? 10 to 100 seconds, something like that. But these are all separate groups with different treatments. So maybe A would probably be better, right? Because treatment 1 and 2 are separate. 2 and 3 are separate, right? They're not connected. So 8, I would say A. Okay, 9. The illustration above is a model of a typical beetroot cell. Based on the experimental results, which of the following best represents the effect of acetone on the permeability of cellular membranes, right? So basically it said that acetone caused damage to the permeability of your cell membrane and your cell released a bunch of the beta cyanide, right? Right, essentially, we know that the image has to have beta cyanide within your cell before treatment, and then the beta cyanide should move to outside of the cell after treatment, right? Only a couple of them you see that actually have. There's only a couple here that actually have that, right? But the second part of it is that it affects the permeability of your cellular membrane, right? So right, you can see that, right, your cell wall is fine, right, but it did affect your cell membrane. and your vacuole right here. And remember, your vacuole is also made of cell membranes, hence the damage. Yeah. Number 10, student analyzed the data from the investigation and concluded that the estimate of the mean of one treatment group was unreliable. Which of the following identifies the treatment group most likely to have provided an unreliable estimate of the mean and uh, and correctly explains why it uh, appears unreliable? Right. So, okay, if we look at the data, let's see. If we just look based on the SEM values, right? This guy is the greatest value, right? It has an extremely high SEM. Probably 2 is unreliable. Why? The absorbance value that you got is 0.125, but it's literally, right, 2 standard area of the mean is 0.1, right? So that means if you look at this graph that we have had, right, your error bar is almost as big as your actual absorbance value, right? Which, I mean, um, is kind of, it means that it's like 0.125 plus or minus 0.1, right? Not a very, doesn't seem like it would be a very good value. Treatment group 2, right, has lower than expected mean absorbance and the largest standard error of the mean, right? Everything else, 3, 4, and 5 didn't seem like there was anything wrong with those. Right, 3, 4, 5, right? 5, right? Even though you see a huge difference, right? It has a very, very small SEM value, right? So it means that whatever results that you got were not too bad. Okay, based on the data from the investigation, which of the following is the best scientific question about organisms living in water that is polluted with organic solvents, right? Um, Basically it's saying what is going to be happening, right? What question can we ask? What question does this experiment really answer, right? Do organisms without pigments have a selective advantage in polluted environments, right? We really did not test this. We did not look at this question at all, right? Okay, part B. Will organisms living in polluted environments exhibit detrimental effects from an increased permeability of their cellular membranes, right? This is exactly, right, the question that we are kind of looking at. Yeah, right. If pollution does cause a change in the permeability of your cell membranes, right, the question then is what is going to happen or how is this going to actually affect my organism, right? Organic solvents from the environment become incorporated into the cell membranes of organisms living in polluted environments. This definitely does, results do not show anything remotely related to that guy. Should governments place more stringent restrictions on the release of organic solvents into aquatic ecosystems? Yeah, not the question that this experiment is answering. Okay. 12. Within a forest ecosystem, there is a large amount of diversity among members of a warbler species. Of the following stages of meiosis illustrated for a typical cell, Which contributes most to diversity among the warblers? Okay, so meiosis, right? Um, what are the sources, right, of genetic variation that you get during meiosis? You get crossing over, right, during prophase one, right, and then as a result of crossing over, right, you have independent assortment, right, where depending on how your um, homologous uh, pairs line up, right, you can end up in um, a wide variety of daughter cells, right? So that's independent assortment, right? So which one of these shows where um, diversity can be genetic diversity arises, right? So we see here, right? I don't know if here crossing over has occurred, right? And then now their orientation at your metaphase plate is going to be what matters, right? Anaphase, whether it's in meiosis 1, meiosis 2, right? Um, no effect on genetic diversity, really, right? Um, what does affect genetic diversity is what gets pulled apart, yes, right? But anaphase, uh, before you get to anaphase, right, your diversity has already been kind of created, right? And then, I don't know what D is. D is not meiosis. Okay, next one, number 10. In an experiment, 100 mice were released into a field which no other mice had access. Immediately after their release, a representative sample of mice was captured, their fur color was recorded, and they were returned to the field. After 20 years, a representative sample of mice was captured again, and the distribution of fur color was again recorded. Okay, so what do we see? Initially, we had what? Majority brown, right? Brown was obviously the most favorable by a lot, right? Fur color among your mice, right? Now, what did you see occur, right? Essentially, you have a shift, right, in your distribution or your fur patterns, right? And after 20 years, the majority, right, or vast majority of your mice are actually going to be gray, right? No longer brown, right? Um, and they are gray, right? Your brown, which used to be 80%, now is only 20%, right? So something about being or having brown, right, um, did not no longer carried an advantage with it, right? Okay. Which the following could best explain the change in the fur color distribution as shown in the table above, right? So whatever we know that gray, right, has a selective advantage, right? And uh, yeah, okay. So the allele for black fur color is unstable, and over 20 years, most of the black fur alleles mutated to become alleles for gray fur. Again, I don't know how this data would support that quite bizarre statement. B, the field was primarily composed of light-colored soil and little vegetation, affording gray mice protection from predators. Yes, right. That is probably possible, right? Um, if you went from like a dirt road or I don't know, something that was an environment that was brown to something that was closer to gray, right? Then now the mice that are closer to gray have a selective advantage, right? It's easier for them to hide from their predators, right? So that is possible, right? And that would explain, right, a change. Sexual selection led to increased mating frequency of black and brown versus gray and brown. Okay, not exactly sure what that would happen. If sexual selection led to uh, um, like this happening, yeah, what would be the possible consequence? Would it be a change in distribution? Um, Not necessarily. More like you would see possible start of future speciation. Okay. Gray mice were harder to catch and so were underrepresented in the 20-year sample. Yeah, possible, not necessarily. Okay, 14. Liver cells manufacture glycoproteins while adipose cells store fat. Which of the following subcellular structures is likely to be more prominent in liver cells than in adipose cells? Okay, so which one of these um, is related, right, with glycoproteins? Right? Nucleus, no. Golgi apparatus, yes. What does your Golgi apparatus do? Remember, it takes, right, anything that is meant for export of your cell, right, or products of your translation, right? And so what happens is that your Golgi is going to take, right, your protein or whatever you just made, it's going to modify it, right? Glycoproteins are like a little tag, like a name tag. Hi, my name is whatever, right? And it helps like identify, right, what type of cell that is. What is this cell? Yeah. And then they'll package it in a nice little vesicle, send it off, right? Cytoskeleton, again, no reason why you should have more cytoskeleton in a liver cell compared to an adipose cell. Um, Plasma membrane, again, same thing as your part C, right? Why would it necessarily have more? Doesn't make sense, right? And remember, always, it's always applicable, right? The picture of like a typical cell that you see, again, you don't really need, I mean, you won't really have kind of like one of everything, right? A lot of times, like based on the specific function of that certain cell, right, you're gonna kind of see like more of a certain organelle, right? So, for example, like you would expect your mitochondria to be especially abundant in a muscle cell, and that is what you do see. Okay, intravenous solutions administered to patients are normally isotonic. Which of the following is most likely if an IV or distilled water is administered to a patient? Right, so accidentally you hook them up to distilled water, right? So what's gonna happen, right? You're basically putting it in water, right? Your cell is here, right? So we have high concentration, low concentration, right? So when you have high versus low concentration, right, what's going to happen, right, due to what principles of your osmosis, right? What will occur, right? Your water is going to enter your cell, right? And as water enters your cell, what happens? Lysis, right? Or your cells burst, right? So essentially, if you stuff an IV of water into someone's bloodstream, right, um, their blood cells would uh, start undergoing lysis, right? Meaning they'll just pop, right? Because water is flowing inside your cell, right? So which one is that? Okay, number 16. Diagram above represents a typical rod-shaped bacterium. Which of the following best describes a feature shown in the diagram that is unique to archaea and bacteria? Okay, so what feature do archaea and bacteria have, right? Your two domains have in common, right? Organism is surrounded by cell wall. Yes, true. Contains ribosomes. Again, true, but it has nothing to do with representing, right? Either archaea or bacteria, right? Organism does not have a nuclear membrane surrounding its genetic material, right? Yes, that is also true. We have naked DNA, right? or we have no nucleus. Right? Okay, organism is not capable of making or providing itself with ATP. Um, okay, hmm, which one, right, is unique to archaea and bacteria, right? Remember, why is eukaryotes, right, called eukaryotes? You being real, right? So, main difference between prokaryotes, or sorry, uh, yeah, what is a feature that they both have that is unique to them? I would go with does not have a nuclear membrane. No nucleus. Okay, which of the following best supports the claim that organisms of different domains share a common ancestor? Okay, photosynthesis occurs in chloroplasts, and the citric acid cycle occurs in mitochondria. Okay, cool. Doesn't really tell me anything. Glycolysis occurs in both prokaryotic cells and eukaryotic cells. Yes. Right. Remember the fact that you have glycolysis occurring almost identically, right? Essentially identically, right? In prokaryotes and eukaryotes, right, is a strong piece of evidence to indicate that yes, at one point in time, right? Um, what is it called? Um, at one point in time, they were right. They had some sort of shared ancestry, right? Introns are present in eukaryotic DNA, but not in prokaryotic DNA. Not really relevant. Errors in DNA synthesis provide some of the genetic variation uh, in a population. Yes, that is possible. However, very unlikely. Best answer is B. Okay, so we have population changes over time in predator and prey, right? And this is like one of those classic examples, right, where you can kind of see how the prey population and the predator population move together, right? Which makes sense because if you have like a fox and a hare, right, um, the more hair you have, right, or the more like little bunnies that you have, right, the more coyotes or foxes that you can support, right? So the numbers will go up together. However, if, for example, my predator's number shoots up, right, then now your prey is at increased risk of predation, right? So your prey is going to drop back down as your prey values, right? The individuals, yeah, those go down, right? Then what's going to happen to your predator, right? They just lost a huge chunk of their food source, right? It'll also go down, right? So you kind of see this like back and forth, up and down relationship. Okay, which of the following data would be most helpful in predicting possible disruptions to the relationship between the predator and prey, right? So time of day doesn't really have to do with it. Average birth weights of predators because heavier offspring will begin hunting at younger age. That is pretty cool. Numbers of additional predator species because additional predators will increase competition for prey. That is important to know um, because, again, right, who is this predator, right? Who is it um, competing with? And then 18, prey metabolic rates because predators are less successful against prey with higher metabolic rates. I don't know if that's true, doesn't sound right, but best answer for this one would be C. Okay. The three spines stickleback is a small fish found in both marine and freshwater environments. Marine stickleback populations consist mostly of individuals with pronounced pelvic spines, as shown in Figure 1. Individuals in freshwater stickleback populations, on the other hand, typically have reduced pelvic spines, as shown in Figure 2. Okay, so um, the pelvic spine, right, that you see, right, marine stickleback versus freshwater stickleback, right, as represented in Figure 1 and Figure 2, phenotypic difference between marine and freshwater sticklebacks involves PITX1, a gene that influences the formation of the jaw, pituitary gland, and pelvic spine. Enhancer sequences upstream of the PITX1 genetic locus regulate expression of the PITX1 gene at the appropriate times and in the appropriate tissues during development. Previous studies have found that a mutation in the hind limb enhancer interferes with the formation of a pronounced uh, pelvic spine. Okay. Which of the following best describes how sticklebacks in the same population with identical copies of the PITX1 gene can still show phenotypic variation in the pelvic spine character? Um, so here, your enhancer sequence, right? You have a mutation in your enhancer sequence, so that's gonna affect the transcription, right, of this particular gene, right? Um, they're saying that how come you can have some sticklebacks, right, in one population, that right has phenotypic variation, right, in regards to your pelvic spine, what it looks like. And you see PITX1 gene is carried on different chromosomes in different individuals. Uh, that cannot be true, right? Expression of the PITX1 gene is affected by mutations at other gene loci, right? Is that possible? Yes, right? Other genes can affect, yes. Genetic code is translated differently in males and females. We know that is not true. Subcellular location of the PITX1 gene changes when individuals move to a new environment. I'm not exactly sure how that would explain, right, the situation that you have here. So 19 is just going to be B. Okay, 20. A mutation that affects PIT1 gene function in all tissue types is most likely to be at which of the following genetic loci? Right? So, in order to influence this gene, which one is going to be the best, right? Enhancer, you can still get a pelvic spine, right, by having a mutation within your enhancer. If you have a mutation in your promoter, that is probably going to be much more significant, right? So 20 is D. Okay, in sticklebacks, which of the following is most likely to occur if a jaw enhancer is disabled instead of the hind limb enhancer? Which one was the freshwater? Okay. Let's go through. The jaw and a pronounced pelvic spine develop normally because the PITX1 gene is expressed in both developing tissues. It doesn't explain what we're asking for. Neither the jaw nor pronounced pelvic spine develop normally because there is no PIDX1 gene expression in either developing tissue. Is that what you expect to see? Probably not, right? If your jaw enhancer is disabled instead of your hind limb enhancer, right? How would that occur? Okay, the jaw does not develop normally, but a pronounced pronounced pelvic spine does develop because the PIDX1 gene is expressed in the developing pelvis, but not in the developing jaw. Perfect. D. 22. Okay, which of the following describes a possible selective mechanism to explain why freshwater sticklebacks typically have reduced pelvic spines? Right, so, how come, right, pelvic spine is much more significant in your marine stickleback? Well, it probably just says that having a larger pelvic spine is useful, right? It has a selective advantage, right, in your marine atmosphere, right? But only in your marine atmosphere, meaning it does not have that same effect in freshwater. Right. Okay. Reduced pelvic spines increase the likelihood of escaping predators in freshwater environments, right? So yes, if your pelvic spine, a reduced pelvic spine gives you a selective advantage, then it should explain it. A works. Reduced pelvic spines expand the range of foods that can be used as sources of energy in freshwater environments. I don't remember exactly what pelvic spines do, but this doesn't seem to be right. Usually they do give you some sort of explanations, but yeah. Reduced pelvic spines invisible to potential mating partners in freshwater environments, right? I'm not exactly sure um, what that would do. Reduced pelvic spines allow individual fish to pass through narrow waterways on their way to marine environments. Cool, unrelated, right? Okay. Hmm? Okay. 23. Hydrangea macrophylla is a species of plant with blue or pink flowers. Flower color in Hydrangea macrophylla is thought to be determined primarily by soil conditions rather than by inherited differences. Which of the following will provide the most direct evidence that flower color in Hydrangea macrophylla is due primarily to soil conditions? Okay, so if you quickly go through it, comparing DNA sequences now, um, doing a cross, not really, right? They want a direct evidence, right? So it's going to be C, right? Do an experiment. Grow your cuttings um, under highly controlled conditions, right? All of your conditions, besides of your independent variable, have to stay constant, right? And then measure your pH. All right. So based on your pH of the soil, right, you're gonna have different heights of your plants. Okay. Based on morphological characteristics, the phylogenetic tree above has been developed for species 1 through 4. DNA sequencing has recently been completed for a particular gene found in all four species. Sequencing will provide additional information for answering which of the following questions. Is species 1 the common ancestor of species 2? No, right? Common ancestor would be down here, not right here. Does species 1 contain the greatest number of vestigial structures? They're trying to say, like, is it the oldest one? Not necessarily, right? Yeah. Are structures 3 and 4 the most closely related? Yes, they are. Should the positions of 3 and 4 be reversed? Remember when you switch your clade, if I put 4 and 3, mhm, 25. Um, oh, sorry. 24 was C, right? The transmission of information from sensory neurons to interneurons typically involves. Okay. What? How do we submit or transmit something from sensory neuron to interneuron? Right. Typically, what'll come after neuromuscular junction? Not necessarily, right? Um, exchange of electrons between specialized proteins embedded in the plasma membrane of the sensory neuron to the plasma membrane of the interneuron. No. Release of chemical messengers into the space between the axon of the surgery or sensory neuron and the plasma membrane of the interneuron. Yes. Transfer of a phosphate group from ATP to the sensory neuron. No. Flow of protons down electrochemical gradient through a good gated channel between the sensory neuron and the interior neuron. I'm not sure. Okay. Brain coordinates the circulatory and respiratory systems of the human body. Control of breathing, for example, involves neural pathways among the structures represented in the figure above. One important stimulus in the control of breathing is an increase in blood CO2 concentration, which is detected as a decrease in blood pH. Which of the following best describes the physiological response to an overall increase in cellular respiration in the body? Okay. So if you look here, right, your heart sends a signal to your brain, which then turns it to your body, right? Sends it to your uh, lungs, rib cage, and diaphragm, right? Um, right? So signals from the brain is going to also signal your lungs, rib cage, diaphragm, also will signal your heart, right? So it's literally like right in the middle. Oops. Okay. In response to depleted blood CO2, pH sensors send signals directly to the rib muscles, right, resulting in an increase in the rate of CO2 uptake by the lungs and decrease. Why would we want to raise CO2 levels? Because we have low, right? But does it work? Okay. Um, yeah. Obviously, right? Your pH sensors don't. You don't have a direct connection from your lungs to rim cage and diaphragm to your heart, right? So A doesn't work. Not based on the image they showed us, anyways. B, in response to low blood pH, pH sensors send a signal to the brain, which then sends a signal to the diaphragm, resulting in an increased rate of breathing to help eliminate excess blood CO2. Right, that's exactly what's happening with B. Okay, so 26 is going to be B. 27. Um, Thyroxine is a hormone that increases metabolic activities within various tissue targets. Low levels of circulating thyroxine trigger the secretion of TSH, thyroid stimulating hormone, from the anterior pituitary. Okay, low level of circulating thyroxine trigger the secretion of thyroid stimulating hormone from the anterior pituitary. TSH secretion then stimulates thyroxine production and release by the thyroid gland. Increased level of circulating thyroxine inhibits further secretion of TSH from the anterior pituitary. Based on the information, what do we know about TSH thyroxine loop? Okay. What is this? OT, um, yeah, so basically, right, if you have low levels, right, you secrete TSH, right? And then what does TSH do? It makes you produce more thyroxine, right? If you have too much thyroxine, right, you're going to block secretion of TSH, right? And because you block TSH, right, you're not making any thyroxine, right? So which one? D, right? You're maintaining homeostasis, right? The correct level of thyroxine. Okay. Stanley Miller apparatus demonstrated that organic molecules could assemble spontaneously in an environment lacking free oxygen and containing water, methane, and ammonia in the presence of an abundant energy source, such as an electric discharge. The research was considered supportive of the organic soup hypothesis, which states that the primitive atmosphere provided inorganic precursors from which organic molecules could have been synthesized in the presence of an energy source. Based on subsequent research, the primordial atmosphere was determined to contain less methane and more carbon dioxide. Um, the new data about the composition of the early atmosphere had which of the following effects on origin of life hypotheses, right? So the actual right origin, right, is a little bit different. Um, so essentially, what do we need? Miller's work was discarded as not relevant to an origin of life hypothesis. Not true. Work was used to refute the new hypothesis about the composition of the atmosphere. Again, depending on what proof they gave, right? You can't just kind of refute it, right? The organic soup hypothesis was abandoned and is no longer being supportable. Also not true, right? D, new organic soup hypothesis was proposed to account for the new data about the atmosphere. 28th best answer would be D. Only answer really. That would work. Okay, ascorbic acid is an organic molecule necessary for the health of plants and animals. Majority of animals, including most mammals, synthesize ascorbic acid from organic precursors, but some primates are unable to synthesize ascorbic acid and must instead acquire it from dietary sources such as certain fruits and vegetables. The L-gulonolactone oxidase, GULO, gene encodes an enzyme that catalyzes a required step in the biosynthesis of ascorbic acid. Most mammals carry a functional copy of the GULO gene, but some primates carry only a GULO pseudogene, which is a non-functional variant. A comparison of GULO genes and GULO pseudogenes from different animals can provide insight into the evolutionary relatedness of the animals. In Table 1, selected members of some mammalian groups are listed, along with an indication of their ability to synthesize ascorbic acid. Table 2 shows an alignment of amino acid coding sequences from homologous regions of the GULO genes and GULO pseudogenes of the organism tested in Table 1. Okay. So you can see, right, elephant, mouse, lemur, they produce, right, ascorbic acid. Orangutan, chimpanzee, humans, we do not, right? So just like, if you think about it, right, how do we get vitamin C? We are not able to make it ourselves, right? So we have to get it from our diet. Right, so this is just the sequence, right, that you see. Okay, 30. Comparison of DNA sequences in Table 2 suggests that a functional GULO gene in lemurs can have a G, C, or T at position 21, but only a G at position 22. Which of the following pairs of prediction is most helpful in explaining the discrepancy? Okay, so 21. Okay, so they're basically saying that if you replace it, right, if you replace, right, whatever is at 21, right, it still works, right? Because it says in lemurs you can have a G, C, or T, right? So it really means that whatever you have at 21 doesn't seem to be super significant, right? At 22 though, it has to be G, right? Why? Substitution at position 21, basically you have no change, right? Is it possible that you get a different amino acid? Maybe. Oh, sorry. Why did I press that? Possible, like possible, right? Part 2. Substitution at position 22 would result in premature stop codon or an amino acid with different biochemical characteristics, frame shift producing an inactive protein, no transcription of the gene, right? So essentially, right, what is something that could, or of these, which is most likely to occur, right? It's a substitution, so we know C will not work either, right? How would this affect transcription? No, A is the only one that works. Okay, which of the following is the most likely effect of the mutation at nucleotide position 7 in the GULO gene of humans? 7. Okay. Mutation results in the deletion of the gulo gene, so no polypeptide can be translated. Right. One mutation at 7 is not going to cause it to, um, like your whole gene to just be deleted, right? The deletion of the single nucleotide causes a frame shift, changing the primary structure downstream of the mutation and resulting in non-functional protein. Yes, very likely, right? Because what is a problem with point mutations such as like insertion, deletion? It causes frame shift, right? So 31 must be. Okay. Lemurs are primates that live on the island of Madagascar off the coast of Africa. Lemurs have a functional gulo gene and are able to produce ascorbic acid. However, primates that live in other places have a gulo pseudo gene and are unable to produce ascorbic acid. Which of the following best explains the genetic variation among primate species, right? It's like food sources on the island of Madagascar were deficient in ascorbic acid, which caused the primates to migrate to the mainland, where ascorbic acid rich foods are more widely available. Okay. First part makes sense. Second part not so much, right? Part B. Human activity on Madagascar, including poaching, drove the other primates into extinction on the land. Doesn't necessarily explain this. Food sources where non-Madagascar primates lived provided ample ascorbic acid in the diet, which removed the selective pressure for maintaining a functional gulo gene, right? Yes, that makes sense, right? Why do we not have a functional gulo gene? Because we don't need to make ascorbic acid, right? Even if you're not purposely, right, taking vitamin C as a vitamin, right, through just your everyday food consumption, right, you're getting a good amount of vitamin C. Which of the following phylogenetic trees best illustrates um, the point at which the mutation in the gulo gene most likely occurred during the evolutionary history of this organism? Right, so which one are the three that does not have the gulo gene? Right, so orangutan, champagne, humans, right? Those guys are going to be together. They all have no gulo gene, right? Um, Elephant is definitely going to be your out group, right? Because they're non-primates, and then you're going to have lemur kind of in that same clade. Looks like this, right? 33A. Right? Okay. Okay, the cells lining the respiratory tract of pigs have receptors for both avian and human influenza viruses. Based on the model above, which of the following best describes the origin of a new strain of human influenza virus, right? Um, Essentially, this is how, right, a lot of viruses do kind of originate, right? Okay. So what happens here, right, in this image, looks like both, right, both avian influenza and human influenza, they infected you, right? And you basically created a new strain of virus, right? So let's go through the answer. New viral strain inherited a mixture of genetic material from both avian influenza virus and human influenza virus, right? That is exactly what happened, right? And because of that, Okay, yeah. So 34A, right? Both infect, right? And then within your cell, right? It kind of gets mixed up, and now you have a new strain, right? Okay, 35. Okay. In the RNA world, for the origin of life, the first protocells or protobionts relied on RNA, not DNA, for information storage and transmission. Which of the following could best be considered evidence in support of the RNA world molecule? Right. So what supports this? Information storage. Okay, so let's see. DNA is more stable than RNA. Yes, but that is true, but I don't know how that would be relevant here. Some RNA molecules in contemporary cells have catalytic function. Yes, that would be useful, right? RNA is so complex in both structure and function that it must have preceded DNA. Doesn't make sense and not true. RNA has secondary structure, whereas DNA does not. Also not true. 35 is B. Okay, 36. Which of the following pieces of evidence best supports the hypothesis that birds and crocodilians are more closely related to each other than they are to other organisms? Okay, so let's look at the choices. Quite possibly, right? Um, different niches, no. No. Probably C. Right, it says skulls of birds and crocodiles have an opening that is absent in the skulls of other reptiles, right? So that shows that those two, right, have some sort of shared ancestry, right? You had a change that occurred, right, just for their clade, right, and whoever made the them. Okay, in an experiment, the efficiency of oxygen exchange across the plasma membrane is being assessed in 4 artificial red blood cells. The table above lists some properties of those artificial cells. Other conditions being equal, which artificial cell is predicted to be most efficient in exchanging oxygen with the environment by diffusion? Okay, so remember, what do we like? We like a large ratio when it comes to surface area and volume, right? The greater your surface area is relative to your volume, right? That ratio, it means that you have more efficient transfer, right? Of anything in, out, within your cell, right? So 37, which one of these guys has the greatest ratio of surface area to volume? Your cube. Okay, 38. To test the hypothesis that a particular plant synthesizes storage lipids by using glyceraldehyde 3-phosphate from photosynthesis, a researcher plans to use radio-labeled precursors to track the molecules through the biosynthetic process. Which of the following rescheduled precursor? Which of the following radio level labeled precursors is most appropriate for the researcher to use. Okay. Um, so basically, we want to track what? Um, particular plant synthesize lipids by using glyceraldehyde, right, from photosynthesis. Okay, so what is the source of something that we can track during photosynthesis? Let's look at uh, the first one, I guess. Nitrogen 15, because atmospheric nitrogen is fixed to amino acids by photosynthesis. Yeah, but what happens to the nitrogen, right? It's not moving through your system. Okay, carbon 14, right, labeled to carbon dioxide because atmospheric carbon is fixed to carbohydrates for hydration. Yep, seems to work. Um, sulfur labeled methionine because amino acids are incorporated into lipids during photosynthesis. That's not true. Phosphorus because lipids are stored in plants as phospholipids. No, so B is the best answer. Answer. I don't know why this is best answer. Okay, mutations in DNA are usually caused by chemical or radiation damage to DNA molecules, followed by imperfect repair of the damage. Immediately after this kind of imperfect repair, there may be a mismatched base pair in the DNA. Illustration below shows an example of a mismatch with the relevant pair bases in bold, right? So here you go, right? You have A and C, right? Which of the following best represents the DNA of the two daughter cells produced when a bacterial cell with this particular mismatch replicates its DNA and divides? Right, so what would that look like? What would happen? Your single strand, right, would separate, right, into its two strands. Top strand would get A, bottom strand would get C, right? This guy right here, um, and then what happens at the bottom? You get something like G. 39B. Okay, iron is an essential nutrient required by organisms from the environment. When intracellular levels of iron are relatively high, living cells synthesize an iron storage protein called ferritin. The induction of ferritin synthesis by iron was investigated in rats. Figure 1 shows the results of an experiment in which cellular levels of ferritin protein were measured in the presence or absence of iron and actinomycin D, a drug that inhibits transcription. Figure 2 shows the results of an experiment in which cellular levels of ferritin protein were measured in the presence or absence of iron and cyclohexamide, a drug that inhibits translation. Okay, so we're looking at what happens uh, when you have iron, right, low iron, what happens when you mix it with actinomycin D or cyclohexamide, right? Um, how does it affect ferritin synthesis? Okay, which of the following biotechnology approaches could be used to identify ferritin mRNA in a sample of total cellular mRNA? Okay, um, which is, so you have your full RNA, right? How can we essentially isolate just ferritin mRNA, right? Again, if you know, right, where ferritin is, right, should be fairly easy to find it, right? You can just cut it, right, and then separate it based on size using gel electrophoresis. Okay. Um, In individuals with iron-rich diets, intracellular levels of iron are higher than in individuals with iron-poor diets. Which of the following best explains how organisms regulate ferritin protein levels in response to available iron? Okay. If you have iron present, right? So like if you compare these two, um, your first two bars, right? Where you have, this is your control, you have nothing, and then you added iron, and then nothing else, right? That's the only thing you added. You do see that when you add iron, right, translation of ferritin does go up, right? Now, group 3 kind of shows you the effect, right, of translation blockers, right? However, if you look, you have plus plus, right here, right? When you have both iron and you have cyclohexamide or actinomycin, right, which basically blocks translation, right? You see a drop in your ferritin levels for the figure 2. Right, so what does that show us, right? When you have high levels of iron, you get high levels of ferritin. Why could that be? Translation is inhibited? No. Translation is stimulated by iron? Yes. Right. When you have iron, you also have ferritin. 42. The data can best be used to support which of the following claims about the mechanism for regulating ferritin gene expression. Right. Um, okay. So again, right, this one shows what happens when you block transcription, right? When you block transcription, um, you have you still have a high rate, right, of that. If you block translation, right, what happens? You don't see, even though you increase your levels of iron, you don't see a corresponding increase in your ferritin, right? So it tells you, right, that translation is significant. I think that your ferritin, right? Uh, the translation portion is the part that is being affected, right? So 41 will be B, right? Translation of ferritin is stimulated by iron. Okay, the data can best be used to support which of the following claims about the mechanism for regulating ferritin gene expression? Iron increases ribosome binding to ferritin mRNA. Yes. Right. Iron causes ferritin to be degraded. We know that is definitely not true. Um, iron increases effective actinomycin D. We also saw that that is not true. Right. RNA decreases the ability of ferritin mRNA to bind on ribosomes. That obviously doesn't work. The only one that works is A. Okay, 43. Gene sequences responsible for the iron-mediated changes in ferritin protein levels are highly conserved and are called iron response elements. IREs have been observed in a number of genes involved in iron transport and metabolism. The IRE sequences found in the ferritin gene are found in all multicellular organisms, whereas other non-ferritin IRE sequences are found only in certain vertebrate organisms. Which of the following is the best explanation for the observation? Okay, so let's look through the choices. Ferritin IREs arose in the common ancestor of multicellular organisms, whereas non-ferritin IREs arose later in specific evolutionary lineages. Right? Um, yeah, right. Which makes sense because one is found in all, one is not. Right. Let's look at the other one. Non-ferritin IREs arose at different times in the common ancestor of multicellular organisms. Okay, no. Non-ferritin IREs arose in diverse organisms by convergent evolution. No, already. Ferritin IREs have evolved independently in different evolutionary lineages where ancestors arose. What? Non-ferritin IREs arose in the common ancestor of multicellular organisms. Nope. Right. A. Best answer. Okay. Hereditary hemochromatosis is an iron storage disease that results in reduced uptake of iron into cells. Based on the data, which of the following is the most likely effect of HHC on ferritin protein levels, right? So it's going to reduce uptake of iron, right? So what is that going to do? Or what is that most likely going to do? It's going to affect your ferritin production, right? High levels of iron, high ferritin production, right? But if I decrease my levels of iron, that will in turn decrease my levels of ferritin. Right. Okay. So which answer looks the best? Elevated levels of ferritin protein, we know that is not true, right? If we reduce iron, we will also be reducing ferritin, right? Reduce levels of ferritin protein because there is less iron in the cytosol of affected individuals. Yes. Related levels of ferritin protein because iron stimulates ferritin breakdown. No. Correct answer is B. Okay, 45. The figure above shows a series of microscope images taken over time of ovalbumin production in chick oviduct cells in response to stimulation with estrogen. Ovalbumin protein was detected using a fluorescent dye, as shown by the white areas in the time-lapse sequence. Microscopic images indicate that the ovalbumin is the yeah. Okay, so what do we kind of see? And so once you stimulate with estrogen, right, you can see ovalbumin kind of taking over that white stuff. I'm assuming, right? So ovalbumin, as you, oh, I don't know, this is such a bad picture. Is synthesized on soluble ribosomes and packaged in lysosomes. Not true. Is synthesized on ribosomes bound to the RFER and then secreted directly into the oviduct. Um, yeah, I don't think A and B work. C crosses the endoplasmic reticular membrane as it is translated, then moves to the Golgi apparatus and to vesicles from which it is released. I don't know. You can't really see from this picture. It does kind of look like it, right? Rough yard, right? Vesicles kind of going to the membrane, right? Okay, 46. Freshwater bony fish secrete large volumes of very dilute urine. Many marine bony fish secrete small volumes of concentrated urine. Which of the following best explains the differences in nitrogenous waste excretion between freshwater and marine bony fish, right? So why is it different? Or what are the advantages, right? Probably you had, right, like A says, the differences arose during divergence as fish from a common ancestor face different selective pressures in environments of the different osmolarity, right? So, yeah, those two, freshwater and marine bony fish, right? You probably had a change that occurred at one point in their phylogenetic tree, leading them to have those different traits. Okay, so 46 is A. 47, malaria is caused by several different species of Plasmodium, a protozoan parasite. Plasmodium resistance to the common drugs used to treat malaria has increased in recent years. Not good. In a scientific study, Plasmodium samples were analyzed and blood drawn from a large number of infected patients before drug treatment and subsequently from the subset of infected patients with drug-resistant Plasmodium. DNA sequences of 4 different Plasmodium genes thought to be involved in resistance with drug-sensitive Plasmodium and patients with drug-resistant Plasmodium. Which of the following best supports the hypothesis that pre-existing mutations confer drug resistance? Okay, so what do they say here, right? Um, okay, so basically for 47, um, can pre-existing or how can pre-existing mutations confer drug resistance, right? Um, Again, if you look here, right? Okay. So again, um, best evidence for that. Some of the mutations that occur most frequently in samples uh, from patients treated with antimalarial drugs are detectable in Plasmodium samples collected prior to use of antimalarial drugs, right? So this is not a new mutation that is being seen, right? Okay, 40-50. Insulin, hormone secreted by pancreatic cells, stimulates glucose uptake in skeletal muscles by mobilizing glucose transporter proteins, GLUT4, to the plasma membrane. As depicted in Figure 1, binding of insulin to the insulin receptor triggers an intracellular signaling cascade in which certain molecules activate other molecules in a relay of the hormone signal to cell targets. One outcome of the signaling cascade is mobilization of GLUT4 from vesicle storage sites in the cytoplasm to sites at the cell surface, where GLUT4 allows plasma to enter, allows glucose to enter the cell, right? Because remember, what does insulin do, right? It lowers your blood sugar, right? How does it? One thing that it does is it's going to stimulate your cells to increase their uptake of glucose, right? Okay. In type 2 diabetes, cellular response to insulin is disrupted, and individuals with type 2 diabetes cannot properly regulate their blood glucose levels. In an investigation of the insulin signaling pathway, samples of skeletal muscle were isolated from individuals who have type 2 diabetes and from individuals who do not. The results of several experiments that were performed are shown in Figure 2, Figure 3, and 4. Which of the following is a valid interpretation of the experimental results that explains how individuals with type 2 diabetes differ from individuals without diabetes? Okay. Um, so if you look at this, right, a regular person versus someone with type 2 diabetes, right? Yeah, you have like a difference here. But what is the thing that is most, I guess, different, significantly different? This guy, insulin stimulated glucose uptake, right? So your type 2 diabetic does not take any glucose, right? So something wrong with GLUT4. Okay, 49. Based on the experimental results, which of the following describes the most likely defect in muscle cells of patients with type 2 diabetes, right? So how would your muscles be messed up, I guess? Um, so this is like the little diagram that they gave us, right? Um, if you have a patient with type 2 diabetes, right, what's gonna happen, right? This, both, I guess both sides of this would be messed up, right? Um, okay. So what would you have, right? IRS activation is reduced at high insulin concentrations, right? How do we know? Right, this guy, right? IRS1 activation for someone with type 2 diabetes is extremely low, right? Now, what does that affect, right? Again, right, IRS1 activation is going to mobilize your GLUT4 vesicles in order for, GLUT4 will control glucose levels. Okay, 50. Based on the information presented, which of the following genetic changes in an individual without diabetes is most likely to result in disrupted cellular response to insulin signaling similar to that of an individual with type 2 diabetes? Okay. Basically, like, what can you do to induce something that will... Let's see. Perhaps A. Deletion in the gene encoding the insulin receptor that removes only the cytoplasmic domain of the protein. Okay, trypsinogen is split by the enzyme enterokinase to form an activated molecule of the protease trypsin. Which of the following would confirm that the activation of trypsin is an example of how a positive feedback mechanism can amplify a biological process? Okay, so positive feedback, product goes back, right? Stimulates a reaction, right? A pathway, right? Activated trypsin can use enterokinase as substrate. No. Trypsin produced by the reaction is capable of splitting and activating additional trypsinogen molecules. Yes, right. That is a positive feedback loop right there. Right. Part C is describing negative feedback. Right. Yeah. Okay. 52. Cancer can result from a variety of different mutational events. Which of the following is least likely to result in the initiation of which of the following is least likely to result in the initiation of a cancerous tumor? Okay. Receptor mutation results in activation of a cell division pathway in the absence of appropriate ligand. Possible. Mutation results in the loss of the ability to produce a tumor suppressor protein. That is actually very important, right? Very important. A defect in the cell cycle checkpoint prevents a cell from entering S phase. If you don't enter S phase, right, that cell is not going to divide. You don't really have to worry about a cancerous tumor. So C. Okay, 53. Which of the following statements is true regarding the movement of substances across cell membranes? Right. Ions are unable to move through the phospholipid bilayer because nonpolar tail regions of the phospholipids are hydrophobic. Yes, that is correct. Right. Remember your phospholipid bilayer, hydrophobic, hydrophilic parts as a result, right? Only small nonpolar things, right, can kind of go through your or diffuse across your cell membrane. Do you guys have your answer for these? Yeah, as well. Okay. Then these should be pretty simple. Are there any ones that you guys want to go over, or are you okay with these? Okay. No questions on these? Okay. Okay. Any other questions on anything in general? No? Okay. Um, if you haven't submitted your Google Docs, can you guys please do that by the end of the day? I think there's only a couple of you guys that submitted it. And then if not, spring break is over for Dan Bar, but yeah, enjoy the rest of your weekend.